Hi, I'm Tanvi Jain, and I'm a rising junior at UCSD, majoring in bioinformatics. And over the summer, I interned with the NIH SenNet CUSP program in Durham, North Carolina. And I was mentored by Dr. Cliburn Chan in affiliation with Duke Medicine. And my main focus of the project was working on visualization of SenNet flow cytometry datasets. As an internship overview, I completed the institutional review board, and then after that, I could work in wet lab with the Murdoch lab. And I shadowed in flow cytometry, wet lab, and blood sample processing. But my main focus was computational work in flow cytometry data sets, especially in visualization. So the rest of my presentation will be about flow cytometry visualization. So for the basics, flow cytometry is basically when single cells are passed through different lasers, and then through that, we can analyze different characteristics about cell populations at a single cell resolution. This could include cell size, cell granularity, and then we can also take advantage of tagging fluorescent antibodies onto cells to help us understand what exactly these cell populations are. So the black table here represents what a FCS data set might look like, where each column is a parameter and each row is an event or like a single cell. Okay, now that you guys know what flow cytometry is, single cell senescent cells have different characteristics, and one of them includes immune interaction and attraction. So this is relevant to flow cytometry because this field is still growing and there is no consensus senescent cell marker identified yet, which means we need multiple, making our data heterogeneous and complex. And this may require a lot of flow cytometry markers. And this could include IL-6, IL-8, PI-1, DCR2, and different factors. So now the main question is, how do we analyze up to or more than 20 markers that senescent populations require, as this increases dimensionality, and someone who is not trained in bioinformatics cannot possibly analyze so many dimensions together. So this is why bioinformatics enable data visualization in fluorescent set of flow cytometry is important because manual gating, which is the current way to analyze FCS datasets, requires a lot of expertise, it's very time intensive, and you must be familiar with specific software such as Flojo. So someone who is not trained or does not know a lot of coding may not be able to easily understand their FCS data sets, which is why we need better visualization algorithms in the right environments so that more people can identify senescent cell populations faster and easily. So some of the flow cytometry data visualizations are shown here. And over my course with the NIH and Duke, I mainly worked on the SPADE algorithm. So I'll be talking about how I implemented SPADE. What is SPADE? SPADE stands for Spanning Tree Progression Analysis of Density Normalized Events. That's a really long word, so I'll be breaking it down in the next few slides. So it basically takes your flow cytometry data set, it normalizes the density, it clusters it, and then it represents it as a tree, and then we can visualize it using the gradient colors here, which shows the marker intensity, which we can toggle later on. So the first step is density dependent downsampling. So over here in this graph, the x-axis is as one marker and the y-axis is another type of marker. So we can only visualize two markers at a time. So imagine the number of combinations of graphs you might have if there are up to 20 or more markers. So over here, the highly dense region is an abundant cell type, and the low regions are rare populations. So, most clustering algorithms usually depend on this density variation to identify different cell types. But in contrast, our spade algorithm removes this density variation to then cluster it further. So I implemented that, and density-dependent downsampling removes your outliers. It removes the redundant, repetitive cell events in the denser regions, and it leads to more uniform density. And it also lowers down our events, as we can see, from 50,000 to 6,000 events in this example. If you want to learn more about how I implemented this density-dependent downsampling, feel free to scan the QR code to my GitHub. So once we downsample it, we can now use agglomerative L1 single linkage clustering to then cluster it into different cell populations. So here, each cluster is represented by one color, and one cluster represents one specific cell population. Now that we've clustered it, we can go ahead and construct a tree out of it. So here we take the mean centroid points of each cluster from the previous slide and then represent it as one node. And then we create weighted edges from each node to every other node using the Euclidean distance between the different marker parameters we have. And then we can use figure B to apply a minimum spanning tree algorithm that creates figure C. And this is the topological structure of our spade tree. Now that we downsampled our 50,000 events to 6,000 events, it's important that we use that information. So we have to upsample it back. So how do we do that? So we take our original data set and then we find the nearest cluster in the geometrical space, and then we can label each cell event with a unique cluster ID. So this would upsample it back to 50,000 steps in a less computationally intensive way. And now that we have now we can use the upsampling to calculate the average marker intensity for each marker in each cluster or node. So this is an example of one marker where the more antibodies of marker X that are tagged on cells will be represented by red. So these cell populations have a lot of marker X tagged on them, while the blue ones don't. So now when our dimensionality increases, we won't always have visualization originally. Like over here, we might have overlapping edges or overlapping nodes. So it's important that we strategically place all of these nodes by finding the longest path and placing that on an arc. And then we can use the Fredgerton-Rengold algorithm to place the other branches. And now that we have our visualization ready, we can toggle between different markers from the 20 that we have, and then we can toggle between them and learn more about what kind of cell populations are we dealing with using the visualization. So that was the end of implementing the SPADE algorithm. My second half of the internship was now focused on using AI to label the spade tree that we made for it to make like more biological sense to someone who doesn't know the data set well. So how can we use AI to help us label the spade tree? So we have identified these different cell populations in each node. So now we can use their differences to then use AI like ChatGPT or different ML models in Python to then identify and label these different cell types. So here's a graph that shows the orange, it represents one cluster, and the blue represents all of the other cells. So we can see how each cluster is different or unique, and then use those differences to classify what kinds of cell populations they might be. So I used XGBoost and SHAP, which are ML models, to then find out what features are important to help classify a specific cluster. And once we have that feature importance, we can set different thresholds, and then we can use prompt engineering to put it into ChatGPT, and then we can tell ChatGPT to identify each cell population's cell label. Unfortunately, I ran out of time, and this process still needs a lot more refining. So we could try a lot of different approaches, excluding ML algorithms that I didn't get to try. But regardless, it was a really fun experience, and I thought it was really cool that about how ChatGPT could potentially help us analyze different data sets. But the figure here shows what the ultimate goal was, where we have a spade tree, and each different cell populations are labeled with their biological cell type. So linking all of this back to SenNet, so we can easily put these senescent marker tag PBMC cell data sets into the pipeline, and now people can understand what their data set looks like better. The good news is that my pipeline will soon be packaged into Flowkit, which is Duke's version of analyzing FCS data sets. I've put a QR code to Flowkit as well. So that brings us to the end of this presentation. I learned a lot over the summer, and that's a special thanks to Dr. Cleburne Chan, Dr. David Murdoch in lab, Kay Metis, Elizabeth Martinez, Natasha Solman, and all the NIH SenNet CUSP interns, of course, and the NIH SenNet team, and the bioinformatics department at Duke Medicine. I've put up QR codes to links to my GitHub project if you want to know more about it. And of course, feel free to connect with me on LinkedIn. Thanks.